Um, our third lecture, the third day of the data science school, will be delivered to you by Alexander Petrenko, who have obtained a master's degree in general medicine and currently pursuing a PhD in the Medical University of Vienna. He's also a research fellow in the Medical University of Vienna and predoctoral fellow at CEMM Research Center for Molecular Medicine of the Austrian Academy of Sciences. And he'll give talk on the topic of development of machine learning models for biomedical applications. So Alexandru, the word is yours.
Thank you. I would like to say thank you to the data science school management and organizers. And I'm happy to see you all here, whether it's like you have nothing better to do on Saturday or you just really are into machine learning, whatever the reason is. Happy to chat with you today. As Vladimir said, I'm a medical doctor, not a computer scientist. And many years ago, before switching to the scientific track, I would not guess that I would ever will find myself working with machine learning. But here we are, and I will try to bring this interdisciplinary perspective into the talk in the next around 20 minutes. So to those with who we haven't met before in the previous events, I graduated from the DSS National Medical University in 2016 and then pursued the postgraduate training in general practice, which I switched to scientific track in which I'm still as of today. So in 2019, I started my PhD in systems biology and liver disease in the Medical University of Vienna and the Research Center of Molecular Medicine here in Vienna. And since last year, I'm also a research associate in the same institution where I'm proceeding with my research. If we focus on the previous works around which I will try to build today's talks, this one and also the next one in a few hours, this will be the talk about gene signatures in a specific condition which is called portal hypertension, and we'll also touch a little bit about this today. And the key work, I think, for today's meeting is this assessment of portal hypertension severity using machine learning models, around which I will build the story. How can you apply in similar applications your machine learning skills? So let's jump to that. And if I want to focus on something today, I would focus on two things, which is what the clinicians want in the clinics and where we are now at this moment, what we have in this example of this work I just mentioned. So on this picture, it is our campus, is our hospital, which is considered quite a large hospital. And as you can imagine, in large hospitals, there are many patients. And for each of these patients, usually we have quite a good coverage from the data point perspective. So we know where they visit, we know how old they are, what is their biological sex, and we also know their parameters. So the reason why they were appointed to the hospital. And better things can be achieved in the clinical medicine with the usage of the machine learning using all I just mentioned. And to build a good thing, I think we should focus on what the clinicians actually want, because they are the experts and they are seeing the patients and they probably have a good guess what are the gaps in this field. And usually they want better accuracy, maybe more accessible tools, so more doctors can serve the patient better without specifically or necessarily being in a large hospital with good equipment. Maybe some new biomarkers or any metrics that could lead to the better healthcare, patient care, and finally more suitable tools for clinical research or clinical trials. And I think the whole story which we developed was built around that. So as I mentioned, in a hospital, there are many data points physicians can add to the medical systems, to the patient's history. And on the left side of the slide, you can see some of them. So there could be data sets with very basic DNA, RNA, protein, whatever readouts clinical medicine uses for diagnosis with FDA or EMA approved panels. They can be patient data sets with just very basic string-based or free type-based readouts, like what is the reason, what are the complaints of the patient, what is the history of the disease, some blood tests, some common instrumental tests, and so on. And finally, what we don't actually use so much into the clinics, but what many patients have today is the equipment not specialized, but still monitors which can track their health status, whether it's physical activity or cardio monitors or other digital devices that they can bring. And of course, having all of this is very um, suggest an approach to switch to the analysis of these things, whether it should be basic statistics we're using for generations or something more novel, such as machine learning. And finally, why would we need all of that? Because in principle, that could lead to better patient care, such as personalized medicine, when a specific patient receives specific therapy or dosage of a specific therapy based on the unique combination of the blood and genetic markers, for example, or can even lead to the better outcomes or more economically reasonable therapy in each specific case. And of course, there are many formats. Like this is what's mentioned here is just an example. We also have a lot of histology, which is nowadays being scanned in large hospitals and being analyzed on computers, which also allows us to hypothesize some computer vision applications, which are being actually uh, actively developed nowadays, imaging and so on. So what can we do? And this will be actually the key part of my talk based on our publication. Jurgi Radius, my colleague from the same research center of molecular medicine, and I published a study a few years ago in the journal Hepatology, which is considered an important journal among clinical doctors, on a problem of portal hypertension severity and how to identify that with machine learning. And I will just quickly bring you to this topic. So when we talk about the portal hypertension, I mean this specific vessel, which you can see here on the left portion of the slide, a portal vein which brings blood inside the liver. So where the cells can exchange with the blood, with nutrients, with whatever cytokines or cell relevant factors are being secreted there, and also secrete these factors back until finally the blood will leave the liver and go back to this general circulation. Now, as you can imagine, if we have a portal vein and we have a condition called portal hypertension, it meaning that there is a specific hypertension or elevated pressure inside this vessel, which can somehow impact the physiology of the patient um, um, or even the course of their disease. And you will be absolutely right. And many chronic liver disease patients have this condition called portal hypertension. There are currently no targeted therapies for that. There are non-selective treatments, which can be either effective or not. And unfortunately, this condition is quite life endangering, meaning that it can lead to um, life-threatening complications such as varicose bleedings, where patient, for example, can die of this severe bleeding. And the problem is there are not many centers in the world which can diagnose that. Like, what is the pressure inside the portal vein? The procedure itself is invasive, meaning that physicians specifically trained using quite expensive equipment use catheter to reach portal vein to measure the difference in the pressure inside that with stimulation, without stimulation. And as you can imagine, in many centers and in many countries, they don't have such equipment or such specialists. For example, in Ukraine, I'm not aware of any center performing that. And what we thought many years ago, like when we started this project, that it would be a great approach to have a non-invasive test, maybe machine learning based on the common parameters that can help physicians to diagnose this condition, to predict which patient might have or might not have this portal hypertension. And this is how it started. There are, of course, gold standards for non-invasive testing, which are not indirect, as well as our approach. For example, ultrasound-based, but still there is a high need of more accessible tools because even the ultrasound-based is not accessible everywhere where we would like to have it. So the question was, is it possible to predict the condition called clinically significant portal hypertension or the specific gradient of portal pressure more than 10 millimeters of mercury and severe portal hypertension, 16, using common lab-based parameters in patients with compensated advanced chronic liver disease. And when I refer to advanced chronic liver disease, this is another way to say that the patient has cirrhosis and compensated, meaning that at this time the patient was stable. However, they can occur decompensation, which can be life-threatening. So in Vienna, as in many centers in the world, we have patient cohorts, patient databases that then being created by physicians. They recruit patients and they record their data points, such as lab-based parameters. Here you can see age of the patients, etiology, various laboratory parameters. And you can see in green this HVPG, meaning that in our case, as we are a big center focused on hepatic hemodynamics, we knew this parameter, which is a reflection of the portal pressure in the portal vein, so like a ground truth. And also, what can you immediately see here that we have some missing data, and this is a really real life situation when in hospitals, whatever good cohort you're aiming to have, you will have this missing data points because either there was not enough of biological sample, serum or plasma, for example, or the kit for diagnosis didn't work, or for another reason. So they had around 500 data points per patient and around 140 patients meeting the criteria we set in our question, meaning compensation and available portal pressure metrics were accessible for our study. So we had different age, sex, etiologies here, some missing data points, and we further reduced it to 133 data points, meaning that we discarded the lab tests and instrumental methodics, which we thought are not that commonly accessible as we would like to have to be. So there were some challenges in this data set. The first one was class imbalance, meaning that there were so normally when we talk about compensated patients with advanced chronic liver disease, we mean that in their compensated stage, many of them don't have too high portal pressure, and as you can see reflected here. So the further you go, the less patient with elevated portal pressure you have. For example, an extremely high portal pressure between 20 and 25 mercury is not in such a big number of patients. And also when we classify them, we're seeing that, of course, with elevated risk, like meaning that somewhat increased portal pressure, but not dramatically, we have more patients than in the high risk. But again, we thought it as a limitation, but also as a good point, like reflecting real life situation in our data set, because this is exactly what physicians would see if they want to use our tool prospectively, meaning that with new patients. The second was nonlinear relationship between the parameters. Here you see some laboratory or instrumental readouts. Doesn't really matter what they mean now, but you can see that when we did Spearman correlation, so the simple correlation between the parameter and the outcome, whether the patient has this elevated portal pressure or not, they can see that actual correlations are not that high on the both ends. So maximum 0.65 or negative minus 0.5 in the best case scenario, which is of course not satisfactory for clinical needs, and we need something more precise. And even though when we did the simple correlation, the top parameters are actually quite valuable in the clinics. For example, the second one, having the second best correlation, is the liver stiffness based on the ultrasound diagnosis, which is a golden standard. But as you can see, it doesn't predict that good in our specific cohort, which is still quite big for such applications. And the third challenges was that arbitrary or let's say hypothesis-driven parameter selection didn't work too well. We selected a certain combination of parameters, which doesn't matter in this regard because in a moment I will switch to the ones we used, and you can see that actually they didn't predict the high portal pressure that well. So the R-squared score was 0.5 in one case, in one case with rich regression, and the support vector machine it was even worse. So again, we needed something less biased, less hypothesis-driven, but more computer assisted in this regard. So we rephrased our question in this context. Can machine learning models classify patient? And when I say classify, I can immediately imagine binary classification task. Yes, no, high pressure, low pressure, as those with significant portal hypertension and severe portal hypertension in compensated advanced chronic liver disease. And second question, which I will only touch in the end, can machine learning predict actually these values, so the absolute values? Not only so the first task is classification, the second is regression, meaning that from certain parameters, can machine learning like predict what is the absolute value of HVPG? So to improve our approach, we did the following things. First of all, we decided to select features based on machine learning, and on my next slide, I will show how we did that. Second, we decided to classify based on cross-validation, which is commonly used in the field, and this is good, even Wikipedia picture on that. When you split your data set into the training and test subsets, for example, the majority of the subset will be training, a small subset will be for testing, and you repeat this iteration k times, 5 times, 70 times, 100 times, There's no limitation for that. And for the performance metric, we would use ROC for each fold, and then we would get the mean ROC from all the folds we use, so meaning from all the iterations. And based on that, we would judge our models. So this is what we did. We classified then our patients as high risk versus low risk because we already knew the ground truth. We had missing values, which I presented in the first slide, and we imputed them. So in the total, we had less than 10% missing values. We discarded those with too much, those laboratory parameters with too much missing values, and the rest we imputed. We normalized the data, and we followed to the binary classification. So for the models themselves, we selected the next ones, logistic regression, super simple, super nice to have, and tried also something more complicated like neural networks, random forest, support vector machine, and extreme gradient boosting. Now the reason for that is, and I think you should always have the reason why people select one machine learning approach or another. We really hope that logistic regression would be among the best predictors. And why is that? Because in such a case, it's very easy to extract coefficients of this regression, and then they use it, for example, in a calculator for clinicians or something like that. And this is exactly what we did, and I will show in the last slides, but just to keep it in mind. So we did cross validations and our ROC as the performance readout. So first we tried to see whether if we just simply add all the parameters, whether the prediction would be anywhere good, and actually was not bad. So we can see that I think the biggest prediction performance was in a support vector machine with our ROC of mean ROC of 0.88. It was not bad. We of course expected it, and validation it will be worse. However, is it really something we wanted? And I think not, because in the real life, for example, when a family medicine doctor or the general practitioner sees a patient with suspicion for portal hypertension, it's very likely they don't have 80 plus parameters available in this family medicine doctor office. So we thought that we might do something better. And as I already told, we wanted to select parameters based on certain machine learning approach. And for that, you use recursive feature elimination. So what does smart phrase mean? That we took all the variables and we, using logistic regression, scored them. And in each iteration, we just removed the one which predicted the high elevated portal pressure the worst. And we repeated this until we reached a small number of parameters. We arbitrarily set it like 5 or 3 parameters. We also had 4 parameters, but it didn't really predict any better than 3 or 5. And we resulted in 2 best performing models. Again, doesn't really matter what these parameters are, but just in case some of you are studying medicine or something like this, this is platelet count, bilirubin concentration, activity of a certain enzyme called cholinesterase, GGT as a marker of a hepatocyte injury, and the APTT as a metric of coagulation. The point is, these parameters were extremely common, maybe except for this cholinesterase. And in the 3P model, you can see cholinesterase is not present, meaning that these are basic parameters which any office of general practitioner can get from a patient and use in their prediction if they want. So we tried only these parameters, and we observed that actually using only them outperformed the whole parameter models. And actually the prediction was quite high already. With logistic regression, we achieved 0.89 as a mean in our internal cohort, which is quite impressive even for clinical applications. So what for the next? We have a reasonably well-performing model in our internal cohort, and of course we wanted to validate it somehow. So this upper panel is from our paper's graphical abstract, just what I told now. So we had this cohort with measurements, we imputed the data and tested the model. The next logical step is validation was to recruit other centers, and we organized a large call for basically all our collaborators, inviting them to submit patient data sets. Of course, if we had the requirement to get patient data sets with 80 parameters, which we had in the beginning, probably we wouldn't gather much. However, because we had only limited number, 3 or 5 parameters as a requirement for the model, we gathered more than 1,000 patients with discompensated advanced chronic liver disease from 7 centers. There were also missing data, but however, we evaluated these models and tried assess their performance. Here I will only show 2 figures related to that. First, this is the centers as panel A, and you can see there is also an imbalance between the classes, meaning that those with non-elevated portal pressure sometimes were more or significantly more as with extremely elevated portal pressure and vice versa. For example, in this Barcelona center, they observed not many patients with normal portal pressure. And if you remember, I mentioned cholinesterase. So as you can see, apparently, and we didn't know this, cholinesterase has been extremely popular in countries like Austria, Germany, Switzerland, Belgium, and in Modena, apparently also in Italy. However, it was not measured routinely in many other centers. So we understood that it's a good approach to use both models, 5 parameters and 3 parameters, because based on which country the user will be, they might not have this cholinesterase chair metrics. And if I wanted to show you only one result, like the final result from the patient prediction cohort, here they are. So this is the prediction for HVPG10, so moderately elevated, and HVPG16, meaning that severely elevated portal pressure, using the 3P model in the merged cohort, so together with the validation cohort. And what you can see here that actually the performance didn't suffer too much. It still was around 0.8 area under the curve. And what is very interesting, it became even better when we combine this with another gold standard, this liver stiffness measurement, meaning the gold standard for non-invasive diagnosis, ultrasound-based for the portal pressure. So we were very satisfied with this result, and as you already seen from the previous slide, it led to the publication. But even more importantly, we built a calculator for that for clinicians where they can just enter these values either from 3 parameter or 5 parameter model and get the probabilities of patient having the severe or extremely severe portal hypertension. Now, I would just say 2 words about regression task. Basically, we use the same models to predict the HVPG. They performed a little bit worse, so we didn't put them as a first point in the paper. However, it's also accessible in them, available in the calculator, and physicians can use that. In January, we reached the threshold of 1,500 unique IPs using our tools. Usually, they are bouncing back, so meaning that people like them apparently. And even though I thought that our tool will actually be used in the countries where the HVPG measurement is not possible or there is no equipment, apparently the United States were among the most, and also obviously among the most active users of our tool, followed by Germany and Italy. So it was interesting for us. And for the last example, because I already don't have much time, I don't want to keep you for too long. This was a poster, and the paper is under review, and this is a little bit different challenge. So in the first approach, we had a lot of patients, right? We have internal cohort of 140 patients, which we then extended to more than 1,000 on the validation. Here, however, we did something with metabolic profiles. Doesn't matter what does it mean if you're not a biologist. Just some markers we found in the serum of some rare disease, which is called portosinusoidal vascular disorder. Now, this is an extremely rare disease, and there is no much centers in the world which can successfully diagnose that. So we had only cohort of 20 patients with this specific condition from several centers, and we used other controls such as healthy volunteers or patients with cirrhosis of non-rare etiology. And we compared metabolites in their serum to understand whether we can successfully predict this rare disease or not. I want to show you this because it's already in the poster on the open access, because I cannot tell too much because it's still under the peer review. However, what you can see from this principal component analysis is that actually the linear way, so linearly, we cannot really separate this cohort too well from the other cohorts we're having here. We achieved some better results using machine learning based on neural networks, but here is, I would say this is the end of what I can tell you openly until the paper is published. But what is important to transfer here, the approach which we used and which is extremely commonly used in rare disease challenges with rare disease cohorts, meaning that leave one out. So this is the name of the approach when you split your data set, not like in our previous case when we had, for example, 20% for testing, the rest for validation, for training. But leave one out, meaning that you train your machine learning model on the whole cohort -1, which is the testing patient, and you repeat it k-fold and see which your results are. You should still consider this method with a grain of salt because it's an extremely overfitting and depends which model you're using. For example, if it's a neural network, you should be very careful with penalizing the model. Otherwise, of course, you will have super high results, which are likely unreliable. But it makes sense as an last approach to find the optimal model, meaning that if logistic regression is functioning for you, great. But even more, if you don't need machine learning and your usual linear relationship are present in the data set, maybe it's even best not to use them in such small tasks. However, this is an example which you can consider. So for takeaways, finalizing the talk, I would like to say that I consider now is a golden age for machine learning in clinical needs because we have so many data points, such big cohorts, like for example, the US veteran cohort, the UK biobank, and so on, that you could easily find your niche, what persona is interesting for you, and try your machine learning applications to improve diagnosis or prediction or whatever you find interesting for your studies. You should consider ethical concerns, meaning that patients should be recruited in the study, willingly sign the informed consent, and the data should not be shared unless minimized under the ethical obligations. Always validate your data, and you need to refer to the domain knowledge for sanity checks. For example, some of the high predicting parameters before we filter out them were those which are very expensive and probably not even possible to perform in many centers in Europe. So you would need to refer to somebody with domain knowledge to ask whether it makes sense or not. What are the best predictors in your case? So with that, I would say thank you. Let's be in touch. Happy to answer your questions. And in case you are in a donating mood today, we have a volunteering donation basket open for the drones and the cars for our army. So you can scan the QR code and send it here and read on our Telegram channel what is it about. So happy to switch to your questions now.
Thank you for great lecture. Yeah, we actually have 3 questions so far. Sharing the screen. The first question is, would be really interesting to see Shapley values applied to initial models before elimination.
I'm not sure we included it actually in the manuscript, or maybe as a supplementary figure, but we access them internally. So if you're interested, you can actually email me, and I am happy to share.
Can you share it with me, and then I can put it like send to the chat where all the listeners could.
Sure.
Okay. Thank you. The next question: impressive work. Can this approach be generalized to other conditions?
I mean, the approach itself, yes, because I think even though the models are quite impressive in the performance, it's not something uniquely done in the world of machine learning, meaning that you can totally use it in whatever application you need with whatever data you need. Importantly, even within this huge cohort we were having, so think about this, patients with compensated advanced chronic liver disease, meaning that inside this big group, there are many etiologies. It can be viral hepatitis, it can be alcohol-related liver disease, it can be metabolic-associated liver disease. And as a question during the peer review process, we actually got this question whether the machine learning model predicts better in one of these conditions or another, and we showed that yes, there are conditions where the prediction is better than we showed in the overall cohort. So I can totally imagine bringing these parameters to just another liver disease etiology we didn't include, and be using the same approach for non-liver disease conditions if you see it fit and you have data.
Right. Thank you. Another question. What is 2D space on the right graph on slide with circles? 2 slides back, starting from the last slide.
Yeah, as I recall, it was just principal component analysis, or really linear relationship between parameters and the 2D embeddings of each data of each patient. So nothing fancy.
Thank you. And the last question: why a set parameter as a feature didn't used for cirrhosis patients?
That's a very cool question. Actually, we also would imagine that because aspartate aminotransferase is a very sound marker for liver injury and has been drastically elevated in whatever decompensation event you can imagine where the liver injury is involved. However, it wasn't predicted that well for some reason. From the liver injury parameters, we only had GGT, which I mean physiologically directly related to the injury, but apparently as that didn't change that much between our patients and also in the validation. So yeah, it didn't score that well compared to others.
Okay, so I think that's it. Thank you for the lecture. Great talk.
Okay, thank you, and see you then in a few hours for the next talk.